Hello everybody, good afternoon. I want to tell you that I am very, very worried, and I have 3 reasons for that. The first one being that is the one the last session, and that in ASA, I'm sorry, I was more worried about all the postprandial effects of everybody and how their enzymes must be working at this moment, and how I am going to keep them alive until the last talk, because, and this is my second worry, I know this is a multidisciplinary group, but basically nobody has talked about what I am going to talk about. So I hope that I can bring the enthusiasm into you to listen and to see if there can be any application to that one. And my third worry is that I have some confocal micrographies, so that either we die because of lack of oxygen and we close the doors and we sleep, or you trust that I tell you what is going to be shown over there.
Trust you.
Thank you. Thank you. Okay, so what I am working on and what I am interested on is on biological models to study different diseases. I am a medical doctor by formation, and then I would like to understand the different how the diseases really develop in the human, basically. And for that, I have been working in the last years in a I think very interesting model, because before that I had been using other models, the cell lines and the animal models, which have given us so much information, but they have a lot of limitations. Basically, and I only list a few, the cell lines, they are limited to a single cell. And because of that, there is no way to study the interaction with the extracellular matrix, which we know is fundamental in the cell signaling between the different types of cells. On the other hand, the animal models, they incompletely reproduce human disease because, among other things, they have different genetic backgrounds. It is increasingly costly to maintain animals in the right settings. And finally, the ethics committees are more and more moving into trying to look for alternatives to do animal modeling, to do biological modeling. So, not so long ago, there was one person, Hans Clevers, that started developing organoids. And I put his definition of organoids as the 3-dimensional structures developed from stem cells and composed of several organ-specific cell types that self-organize along a spatially restricted lineage. Because this might seem so complex, what I always tell my students is that what we do with organoids is take a stem cell and induce differentiation into the tissue where it originated. And basically then you have so many advantages, because you will have the genetics of the stem cell, you will have the architecture of the tissue, and you will have the physiology. I lie a little bit because it's not exactly like that, but they have understood that part. So obviously this development came a big boom in biological models, and it was and is still very much used and published in the main journals. And now we see, or maybe not now, since then we saw the many applications that this kind of model can have. For example, we can do biobanking of patient-derived organoids, and we can use that for precision medicine. And this, because it has the genetics and architecture of everything of the particular patient. We can use this model to study transcriptomics, proteomics, epigenomics, metabolomics, and so on, and it will also give very useful information. We can do gene editing in this model, and we can also use this model to test different drugs for different kinds of diseases. So it is really a very, very powerful tool to be using in the lab. So I am interested basically in intestinal organoids. You can do organoids basically from every tissue in the body. I am interested in intestinal organoids, and I hope I can show you 3 applications that we are using for this development. So what we use, and let's say I put a mouse here, but it could well be a patient, and we take a biopsy, and we have done it, I will tell you. And we take the site of the gastrointestinal tract that we want to use, we want to expand, and we do a little bit cutting and incubating and so on until we get the stem cells, which we put into a gel, a matrigel, and with different kinds of growth factors, we induce differentiation. And in the differentiation, we get a culture, we cut that again, we expand it, and we get what we call here the organoids. They are spherical structures that later we can even open up if we need a directionality. I will show you in a while. But who's going to know what is there? Well, we have to test it. We have to look, in this case, for the different populations that normally we have present in the gastrointestinal tract. In this case, for example, we use gene expression to see that we have enterocytes, goblet cells, enteroendocrine cells, tuft cells, and that stem cells are still are also there. So with this kind of approach, we have seen that we can see in several weeks after inducing the organoid development, the expression of different types of genetic markers, and we can assure that those populations are present in our system. So now, first application that we use this, where we use this approach is to use the infection by Giardia lamblia. So Giardia, and this is a scanner here from our lab, Gabi. Giardia infects the duodenum, the small intestine. And why is it important? Well, in some parts of Mexico, for example, Giardia affects 1 in every 7 children that are under 10 years of age. And this has been seen that they have long-term effects, so it is important to study that for us. Usually, it is a self-limiting disease, and diarrhea is the main symptom in 96% of the cases, but there are many infections that go unnoted because they do not have any symptoms. It is transmitted through cyst-contaminated food and water, so it's very easily transmitted. And the diagnosis is through microscopy, PCR, and antigen detection in serum. Now the problem in Mexico and in other parts of the world is that many times people have diarrhea and they just don't go to the doctor, don't get the diagnosis. They keep perpetrating the infection because maybe they think last night there were too many tacos, maybe the sauce was too hot, and so on, and they don't take care of that. So, but it's important to know about this disease. So what have we done with this system? Well, we took human duodenal biopsies, and we studied first the presence of the different lineages of the cells. And we could see, this is only an example, we could find the enterocytes by labeling the cells with villi, and we could see the tight junctions through antibodies with anti-E-cadherin and ZO1, which are of utmost importance in the intestinal mucosa so that nothing gets in our body, but this is really sealed. Now then we saw that in the tissue we could amplify the biomarker genes of the different cells that I already told you. This is tissue. But with this tissue, we took the stem cells and we started developing the organoids. And this is the extraction of the crypts. We can see day 4 organoids. These are very round. We were very excited. We could get them to split and to multiply to day 7, and then COVID-19 hit our lab. So we had to stop this part, and we continued, but with the mouse model. So what I'm going to show you next is done basically with mice. And so with mice, we were able not only to produce the organoids, but to open them up. Why is it important in a system like this? Because the gastrointestinal tract is a polarized culture. It has microvilli, which we could identify very well through electron microscopy. And these are very important because these are the ones that are exposed to the lumen of the gut. And we infected them with the parasite. You can see here, I don't know if you can see, or you can trust me, about the small giardias that are basically put on top of this monolayer, open monolayer. This monolayer is an air-liquid interface monolayer. It is important because what we could do with that is that we were able to infect and to see, these are the small Giardia trophozoites. It is a protozoan parasite. And you see that it is not completely attached to the cell. Why is that? Because these organoids produce mucus, just like our gut does. But what we could see is that the presence of this parasite reduced the size of the microvilli. And this has very important implications, among other things, because it reduces our area of absorption of nutrients. And if this is done in a child that is less than 10 years of age, and this is chronic, it will produce a malabsorption syndrome. They might be eating, but not getting all the nutrients that they have, and they might not grow as much as they should, and they might have other effects. So we have done that, we have analyzed other things, but because the idea today is just to present the model, I will go to the next example. And this is ulcerative colitis. This is another project that we're doing with organoids, and we are moving further down into the gastrointestinal tract. We are using now the colon, and you might know that ulcerative colitis affects millions of people all over the world, and that to date there is no treatment but a palliative treatment. I mean, there is the production. You can see this is a healthy colon, and this is an ulcerative colitis colon, which is called like this because there are ulcers that are produced on the surface of the epithelium. And so what is happening here? There are several phases of this development of this disease. In a normal colon, there is constant renovation. Actually, the gut is the most efficient tissue that renovates itself. It is constantly shedding cells and renovating. So, but what happens when people have ulcerative colitis? It doesn't matter what the insult is to the gut, but the gut tries to repair. When this insult is perpetrated more and more, then the repair cannot completely restore the beautiful crypts that are the signature of the colon. So with time, there is no way to hold all this tissue, and then damage will occur, and the ulcer will be formed. And before, when they were trying to repair, you saw that could see that cells were trying to migrate and say there is something. Otherwise, I put the other one. There is something stopping here from coming, and then they cannot repair anymore. The inflammatory cells are doing their job, and then they cannot do anything. So what we are doing now in this project is we are trying to see not the way they are being damaged, but what about regeneration? Which cells are the ones responsible to make the regeneration of the tissue, because that is how we can really help these patients. So you can see we have a model of ulcerative colitis, and by adding chemical insult to the mice, and you can see only after 5 days of the treatment, there is a shortening of the whole gastrointestinal system. This is after 8 days of the treatment, and we can see the ulcers that are formed in this system, and we can identify the different cell populations. And these are the enterocytes we can identify with specific markers. And we have done organoids with these different mice. The healthy mice develop big, nice organoids that we can see are also, these are the signs of the goblet cells, that they are polarized. And the ulcerative colitis organoid-derived mice are very different. And now we are in the process of studying this one. So maybe in the next Congress I can tell you about the rotavirus. Thank you very much.
Thank you. We have time for questions. Have you studied Helicobacter pylori?
We have not, because there is another group in Germany that has done it. So in the Max Planck Institute for Infectious Biology in Berlin. And basically, this collaborator is the one that taught my PhD students how to develop the organoids.
Hi. Thank you. I have a question. How similar will be the metabolites produced by that organoid compared with the real tissue?
Well, it has similarities and it has very big differences. The main difference being there is no microbiome over here. So, and you are adding a culture medium instead of adding the nutrients. However, you have the responses that the tissue itself will have in the in vivo normal environment. But it cannot be exactly the same. What some people have tried is to add not the whole microbiome, but select some of the specific bacteria or bacillus that have been identified that are more important for one or the other disease that they are studying.
Thanks for the talk. I don't understand how do you generate the two conditions of organoids. The methodology, you use the stem cells of both the treatments, where the control the treatment and derive on those.
We take mice and divide them into groups. One of the mice are fed water, and the other group of mice are fed DSS. DSS is a chemical compound that makes lesions in the large intestine. So once we treat that for several days, we saw that they produce ulcers without killing the mice. and at that moment we stopped it, and we took the crypts, the intestinal crypts, and isolated the stem cells of the two groups, and then the two groups were grown into matrigel and induced the formation of these organoids.
You can also stimulate to go to the differentiation and generate an stem cell from adult cell, a differentiate.
That is exactly our theory. We think that when there is enough damage and the stem cells of the patient are eliminated, then there is a differentiation of the cells, probably the tuft cells and maybe other cells, and because we have seen the appearance of embryonic markers in some of these of these systems, but we have not been able to identify them in the organoids yet. I mean, this is in progress.
Thanks.
So I'll ask one question about what you can do with organoids. So my understanding is that when at birth, the intestinal lumen is not fully developed, like there's no mucus layer. And so if you were to take like a baby mice and make an organoid from that, could you recapitulate that, do you think, and study the development?
The development of the
mucus layer in
the mucus layer is formed even in the very newborn mice.
Okay.
And the microbiome will start changing it. It is not that they don't have the mucus, it is that from the outside something is hurting the mucosa, and from the inside the inflammatory cells themselves are hurting the mucosa.